Hello, everyone. It's my great pleasure to introduce Marina Picciotto. She is the Charles B.G. Murphy Professor of Psychiatry and also in the departments of Neuroscience and Pharmacology at Yale University. After receiving her bachelor's from Stanford and doing her PhD at Rockefeller, she did a postdoc at the Pasteur Institute. And today you'll have the opportunity to hear about her outstanding and very influential work on largely on nicotinic acetylcholine receptors and how they can be involved in psychiatric disease. She's published over 220 papers, and for her work she's received a number of awards. I'll just name a few, including the NARSAD Independent Investigator, as well as the Senior Investigator Award, the very prestigious Woletsky Award for Innovative Research in Drug Addiction and Alcoholism, and she has been elected to be an AAAS Fellow and also the National Academy of Medicine. Perhaps equally important or impressive about Marina is her incredible service. She serves on a number of advisory boards, steering committees, and award selection committees, and is now the chief editor of the Journal of Neuroscience. And as a mentor, Marina is truly inspirational. She's a mentor to mentors and a role model to all. She's incredibly generous, not only to her own trainees, but I find to all young scientists in the next generation. I remember my very first SFN where I presented a poster on Wednesday morning where Marina was the only person to attend, and I found that incredibly inspiring to this day, and I'm sure she has impacted many of your careers as well. So please join me in giving Marina a very warm welcome.
So awesome. That may be one of my proudest achievements, to be one of the poster attendees on the last morning of SFN. Thank you. That's really cool. So we work on a lot of different behavioral constructs, and the ones I'm going to talk about today are related to anxiety and depression. And I'm going to show data that we have that is perhaps largely pharmacological, but really tries to go back and forth from humans to rodents in a way that perhaps is tractable to really do translational work from basic neuroscience to human psychiatric illness and back again. So I'm going to show you the first slide, which is one that perhaps every one of you has in your slide deck, and it's one that I'm aiming at Steve Hyman because he's actually written on this. And the question is, how do we actually model depression or anxiety in a rodent? And every one of you has seen this slide because, of course, an anxious rodent, a depressed rodent, and a happy rodent all looks the same. And the answer that I have to that question is that you start with a human. Right? So all of the things that I'm going to talk about today actually began with manipulations in human patients. So in the 1970s going into the early 80s, Janesky and his colleagues actually did a number of pharmacological experiments in humans, both first with depressed people with a history of depression, and then in people with absolutely no history of psychiatric illness, where they gave people a cholinesterase inhibitor. So acetylcholinesterase is the primary breakdown enzyme for acetylcholine. So what they were doing was increasing the lifetime of acetylcholine in the human brain and body acutely one time, and they asked these patients, how do you feel? And across a number of studies, which were published in a series of papers, what Janesky and his colleagues were able to do was induce symptoms of anxiety and depression acutely in human subjects, whether or not they had a history. And what does that suggest? It suggests that in some way, during active behavior in humans, if you increase the amount of time that acetylcholine stays in a synapse, we don't know where, that you can induce something that humans endorse as anxiety and depression. So where is this acetylcholine being used? Where is it signaling? Well, of course, we all know that the primary neurotransmitter in the autonomic ganglia is acetylcholine. It could very simply be that people had more signaling in the sympathetic nervous system, therefore felt heart rate increases, felt symptoms of anxiety, although that doesn't really explain the symptoms of depression. We know that throughout the brain, actually I can pace if I feel like it. I did put down my pointer somewhere. Did I put it? All right, I can't paste because I lost my pointer. No, that's not it. Oh, this is it? That's the advancer. I think I have strewn the various paraphernalia of speaking across the entire.
I think it also is.
All right. So this will be my IQ test, because if it is a laser pointer, I will now mistake it for the slide advancer, and I apologize in advance. All right. So yes, we have acetylcholine signaling. It's essential for sympathetic tone. It's essential for sympathetic signaling. But we also know that there are cholinergic cell bodies in number of brain areas, and in fact in increasing numbers of brain areas as we do single cell sequencing throughout the brain. But primarily there's a number of cholinergic brainstem nuclei that are absolutely essential for arousal, and then the basal forebrain complex that are, these neurons are projecting extremely widely throughout the brain, and they're innervating pretty much all of cortex, the thalamus, the hippocampus and amygdala, and in general, these neurons are neuromodulatory. They are, however, activated by stressful stimuli. And this is another old paper. This is, in this case, from rat, and this is a study using microdialysis where extracellular acetylcholine is sampled in the hippocampus, dorsal hippocampus in particular, after a physical stressor. So in this paper by the Shores group, the microdialysis probe is implanted, and then the animal is restrained, and what you can see is that acetylcholine in the dorsal hippocampus goes up as the restraint stress continues. Because of sampling problems, it's likely that this acetylcholine is actually going up concomitant with the beginning of the stress and ending probably somewhere around the end of the stress, and staying high as long as that physical stressor is around. So this is one brain area where we know that stressful environmental conditions are not just activating the autonomic ganglia, but also central nuclei that are important for innervating broad swaths of the cortex. So if this is the case, then increasing ACH signaling may be a model of maladaptive stress response. And the question is, can we take that observation in humans with the cholinesterase antagonist, replicate it in mice, and start to use that perhaps as a model of what acute acetylcholine signaling might be doing in an actively depressed human, and start to take apart where this is happening in the brain, or if it's happening in the brain. So that's where we're going to start. So we had a number of experiments that went into looking at the cholinergic system and trying to evaluate where we might look. But first, we simply replicated Janowski's experiment, gave the same cholinesterase antagonist peripherally, physostigmine, and we asked, can we use first of all, biochemical measures to see if we're really decreasing breakdown of acetylcholine. We can, in fact, use biochemical assays of acetylcholine esterase activity, show that we can reduce it centrally. And we can use these much maligned batteries of stress-induced behaviors to see if there's a change in the activity of these animals. So first of all, we used, in this case, tail suspension. It's the easiest experiment you can do in a mouse. You suspend an animal by its tail. The adaptive response should be probably to continue to try to right itself. If you give an acute injection of an antidepressant that works in humans, the animal will try to right itself for longer. If you increase stress, then the animal will give up sooner. So immobility is a sign of giving up. And when we give this physostigmine peripherally, these animals actually give up more. And this goes along with the ability of the physostigmine to reduce acetylcholinesterase activity. All right, so that's where we're starting. We know that acetylcholine is mediated through two broad families of receptors, muscarinic receptors, which are metabotropic G protein coupled receptors, nicotinic receptors, which are pentameric ion channel containing receptors. They're the ones that we tend to focus on. But we know also that these receptors have to be the transducers of whatever acetylcholine signaling is induced by this pharmacological challenge. So the first question is, can we block these acute effects of acetylcholine in these models of stress-related behavior with pharmacological means? And the answer is yes. If we give either a nicotinic antagonist or a muscarinic antagonist along with the acetylcholinesterase blocker, then we reduce not only the ability of the cholinesterase to induce this immobility, but we have this antidepressant-like response. It looks a lot like antidepressant responses we've seen with other pharmacological challenges. If we put them together, it's a little bit better, but we may have actually maxed out with one or the other, which means that probably acetylcholine is acting it needs all of its receptors. If you take out one of these families of receptors, we don't get this stress-related behavior. All right, that's not really very surprising. If you increase acetylcholine, hopefully you can reverse its effects by blocking its receptors. More important, if we're actually going to call this in some ways a similar response to what we see in humans, we should take a completely unrelated compound, one that works in humans as an antidepressant, and see if it reverses the effects of this cholinergic change. So we took fluoxetine, Prozac. And what we see is that Prozac actually reverses the physostigmine effects very potently. So here's the immobility that's increased when we cholinesterase activity. It's reversed back to baseline by fluoxetine administration. And what this suggests is that this is not simply a reflection of system-level activity that is completely unrelated to the kinds of behaviors that might be important for anxiety and depression, but in fact generalizes across systems that are involved in human depression. So, okay, here we have our pharmacological model. Let's now go in, use what we can in animals to find out where this is happening. This is still peripheral administration. It could still be happening in the sympathetic ganglia. Where actually in the brain is it in the brain? So again, here's where we started. Acetylcholine has actually been evaluated in many different brain areas. There are some brain areas where it changes, but really not very much. Amygdala, for example. You can see that restraint stress increases acetylcholine signaling. It's very, very high at baseline. I will come back to that. It doesn't go a whole lot higher with a physical stressor. The most inducible area that's been seen reproducibly is hippocampus. So we decided to start there. And what we did was first to develop an acetylcholinesterase knockdown strategy. We used AAV2. Now, adeno-associated viruses are very heterogeneous. AAV2 is useful because it infects cell bodies. So if you put something into an AAV2 and you put it in the brain, you know it's going to be in the cells with cell bodies in the brain area that you infuse, okay? And we did a nice rescue experiment where not only did we put a small hairpin RNA that targets the mouse ACHE, we also designed a cDNA with the human acetylcholinesterase so that we could, at the same time that we were knocking down the mouse copy, if we wanted to, replace it with the human copy that was not sensitive to this small hairpin RNA. Okay, so we have a control. And you can see here is that when we knock down the shRNA, we lose this dark staining, which is actually, again, a biochemical measure of acetylcholinesterase biochemistry, of activity of the enzyme. You can see that for whatever reason, this AAV2 really just likes the hippocampus. When we put it in, we don't have very much activity for subregions of the hippocampus. It seems to fill the whole structure, which is probably a plus and a minus. But what you can also see is that when we knock down acetylcholinesterase in the hippocampus more broadly, when we look at exploratory behavior, which is simply whether the mouse is going to go someplace scary or not, that knocking down the cholinesterase locally in hippocampus is sufficient to recapitulate blocking cholinesterase activity in the whole body. There you see that how much a normal mouse will explore a light compartment of a plus maze. That goes way down when we knock down cholinesterase and increase local acetylcholine signaling in hippocampus. Same is true in another model like dark box. All of these are simply where does the animal choose to explore. That's where we started. Now I can show you a number of these other stress-induced behaviors, and that is all of these models that are sensitive to antidepressants that work in humans. Two of them, tail suspension and forced swim, are those same immobility tests that I told you about. But one of them has a little bit more face validity because it's sensitive not to an acute administration of classical antidepressants, but is only sensitive to chronic administration. So let me walk you through this a little bit. What you can see here is that if we put that shRNA and we increase cholinesterase activity only in the hippocampus, we get more immobility across these two models. It's good. When we work on mouse models, there's a million reasons that you can change the activity of a mouse, change the behavior of a mouse, and if the behavior changes in the same way across these models, a little bit more confidence that you're measuring what you think you are. And what you can see here is that if we just use the control, the scrambled shRNA, there's absolutely no effect. And if we use the shRNA plus the human replacement of the shRNA, so this is a control non-selective effects of dicer, any off-target effects, we completely rescue the ability of the shRNA to both increase this immobility and also, in fact, to change the activity of cholinesterase. In fact, we get quite a lot more cholinesterase activity when we replace with the human form. Okay? That's true across these two models of immobility. But here is something where we were actually a little bit more interested. So social defeat stress is something where, and I talked about this with Kay, it's not a super great model for depression because it's really an adaptive response. What happens is that you take a normal, happy, social mouse who likes to explore other mice, and you expose it to a big bully mouse of another strain, and the big bully mouse beats it up for a little bit, you separate them, and you expose the two mice to each other for long enough that the very sweet social mouse now no longer wants to have anything with other mice. So on the training days, and you can do it for 3 days, you can do it for 7 days, you get exposure and learning that there's a bully mouse around, you get the stress response mounting, and then on the test day, you expose this mouse to a brand new conspecific, and you ask, does the mouse choose to go and explore the new mouse, or is it going to avoid the area where there's a mouse? So if we do this for 7 days, what you can see is that on the test day, a normal mouse exposed to 7 days of defeat stress will interact less with a mouse. This is actually not surprising. This is a mouse that has learned that others can actually do it harm. But we did a sub-threshold social defeat. We only exposed the mouse for 3 days to the scary bully mouse. And in that case, it actually is a suboptimal social defeat stress. That's the exploratory behavior of a non-stressed mouse. And this is the exploratory behavior of an animal that has had the acetylcholinesterase knockdown in the hippocampus only, exposed to this sub-threshold level of social defeat. And here's the controls again. If we use the scrambled shRNA, nothing happens. And if we rescue the shRNA with human cDNA, we also don't have an effect of this cholinesterase change. So what that suggests to me is that increasing cholinergic signaling locally in the hippocampus is sufficient to mimic the effect of the systemic blockade that we saw worked in humans, works in mice. And at that point, we can ask, do changes in cholinergic signaling actually happen when people are acutely depressed? So we know that we can artificially increase cholinergic signaling, or we can't, Janowski can, but is this reflective of anything that's actually happening in human depressed patients? So we were very in that we took part with our colleagues in developing a nicotinic acetylcholine receptor tracer that works in humans. We first used it for SPECT imaging, now it's being used for PET imaging. It can be used in awake-behaving animals, but it can also be used in awake-behaving humans. And the way that it works is that here is actually a diagram of the channel that is the nicotinic acetylcholine receptor. In this case, SPECT tracer binds at exactly the same spot as either nicotine or acetylcholine at the interface between subunits of these nicotinic receptors. We can infuse it into the brains of people who are non-smokers. People who are smokers have very efficient competition by nicotine of this binding site, so you don't really see any binding. But if we infuse this into the brain of non-smokers who are either not depressed or actively depressed, we can look at occupancy of this site. All right, and this is one example. This is a control non-smoker. What you can see is, so here's the brain, that's the sort of the nose part. This is a side view, nose again. This hotspot, that's the thalamus. Thalamus is loaded with high-affinity nicotinic acetylcholine receptors. This tracer shows them just the same way they would in a mouse brain slice. Human is a very good model for mouse. And what you can see is here is an acutely depressed non-smoker. What you can see quite clearly is that in this heat map, there is less binding of this tracer. So when my colleagues first saw these images, they said, hey, Marina, very cool. People who are actively depressed have fewer nicotinic acetylcholine receptors. Now, there were a few problems with that. One is that the only human disorder I know of that actually gets rid of nicotinic acetylcholine receptors is neurodegeneration. Neurons hold on to these receptors unless they're dead. So it seemed unlikely to me that an acutely depressed individual would have receptors, but it was possible. The other problem with that is, of course, that this is a competitive tracer. So what does that mean? It means that if there's some level of acetylcholine circulating in the extracellular space, it's going to occupy some interfaces of the nicotinic acetylcholine receptor. If it's low enough, there's still going to be interfaces that are available for the labeled tracer to bind. If instead there's more acetylcholine signaling in the brain of someone who's acutely depressed, there are going to be more interfaces occupied by the endogenous acetylcholine, and now when we add the tracer, there are going to be fewer spaces for this to bind. So we could test that. So here's the data from the SPECT imaging. This is actually also very interesting and was very surprising to us. What we saw was at any brain area that was examined, there was less binding of this competitive tracer to the nicotinic acetylcholine receptor. So that suggested to me this was not hyperactivity of one pathway, but something more broad about either the metabolism of acetylcholine or in some way the biogenesis of acetylcholine that was not dependent on activity of a single pathway. The other thing that you can see is that this was seen in many different cortical areas, but was also seen in midbrain and brainstem structures. So again, not a single set of neurons, which was very surprising to me. The other thing you don't see on here, just for confusion's sake, is that although this is people who have never been depressed, and this is people who are acutely depressed at the time of imaging, people who had a history of depression but were currently admitted, right in between. Kind of cool. All right, so we as neuroscientists could actually help our clinical colleagues by asking for postmortem tissue from the brain bank, Douglas Brain Bank in Canada, where we were able to get control brain tissue, mostly from people who accidents and so on, age-matched and sex-matched non-smokers who died while actively depressed, mostly from suicide, and then age-matched and sex-matched schizophrenic brains as a sort of a psychiatric control. And what we were able to do is then wash out acetylcholine, because you take little slices of brain tissue. We took the anterior cingulate, because it was one of the brain areas that had been sampled in the SPECT imaging, and we asked, if you wash out all the acetylcholine, is there a difference in number or affinity of the receptors that bind to this tracer? And the answer is absolutely not. So in gray matter and white matter, there was no difference in nicotinic receptor number, and I can tell you also affinity. So whatever's happening here, it is not due to loss of nicotinic receptors. So then we collaborated with Irina Esterlis, another of our clinical colleagues, to ask if we could recapitulate the Janowski study. Let's treat these, a bunch of normal controls, with the same cholinesterase antagonist, with physostigmine, and ask if, first of all, do we, recapitulate, see, I failed the IQ test, damn it. Can we recapitulate this finding in individuals who are challenged with physostigmine? And the answer is absolutely yes. This is 5 individuals, each of which is its own within subject control, and they're imaged before physostigmine or after physostigmine, and all 5 of these individuals, physostigmine challenge recapitulated what we saw in actively depressed patients. In addition, we have replicated this. This is in unipolar depressed patients, so the first study that we did. This also happens in patients with bipolar disorder when they are actively depressed. That suggests because bipolar disorder and unipolar disorder do not share an awful lot of genetics, that this is probably not really due to some crazy cholinergic genetic defect in people who are depressed, but potentially something downstream of other defects. And that's really where I wanted to show this to this audience in particular, because when I and colleagues have looked for genes in cholinergic biosynthesis chat, cholinesterase itself, in the available genetic databases, there has not yet been a demonstration that those genes are linked to depression. So it either suggests that this is downstream of some other pathway that is primary, or that this is in some way the consequence of stress signaling is chronically changed. And I want to just tell you one piece of information that I didn't add in here, but this is kind of important. In the cholinesterase knockdown studies, I showed you that we did the control that you could rescue the cholinesterase phenotype by putting in the human cDNA. And I told you we did that one injection at one time. If instead of rescuing at the same moment, we go in 6 weeks later and we put in the human cDNA into the hippocampus of animals that have had the shRNA, we no longer rescue. So there is plasticity also downstream of this out of control, I will say it, out of control, cholinergic signaling that we see in the depressed brain, that we see in remitted patients, that it no longer appears to be sensitive to manipulations of acetylcholinesterase, of rescue of acetylcholinesterase activity in the hippocampus. And that's something we're really interested in. All right. So can we now, we've got this mouse model, can we use it to ask where in the brain is cholinergic signaling really important for these stress-related behaviors? We know that acetylcholine is really released throughout the brain. We've seen even in the human that there's many, many brain areas implicated. But now we can start to go in with tools that we have, that we've developed for many reasons, to examine the nicotinic receptors, for example, and ask, where is this happening? So the first thing I'm going to show you again is pharmacological. We showed actually a long time ago, 10 years ago now, 11 years ago, that if we block acetylcholine receptors, or if we decrease acetylcholine signaling through these receptors using a partial agonist. So as you all know, a partial agonist can actually activate its receptor, but so much less than the normal endogenous ligand that, in a sense, if there's high acetylcholine signaling, a partial agonist would decrease signaling. We could give mecamylamine, again, antagonist, or cytosine, which is this partial agonist, to normal mice, and we can basically increase struggliness, decrease immobility in these tests of stress-related behavior. Okay, so we know that. And the goes along with the idea that if we can increase acetylcholine signaling only in the hippocampus, we can increase these stress-related behaviors. Now, if we decrease nicotinic signaling, even in the absence of an acute stressor, I do think of mice as really kind of chronically stressed. They're a little bit like assistant professors or graduate students. So I'm not sure that we can call them completely normal. However, if you decrease acetylcholine receptor signaling only in mice that have not had other interventions, you can have this antidepressant-like effect. So does this share neurochemical measurements, neurochemical aspects? Just as I was showing you that we can use acetylcholine to reverse the effects of these cholinergic manipulations, can we now combine different agents and see if we can get nicotinic and, in this case, serotonergic agents to work in tandem? So here we have, again, a pharmacological study. Here is this partial agonist. If we use a subdose of the partial agonist, we have no behavioral effect. If we use a relatively low dose of fluoxetine, it doesn't have much of an effect. But if we combine those two subthreshold doses together, we see not only a decrease in this immobility test, but we also see an increase in association with a conspecific in the social defeat test. These two together suggest that somehow there's convergent signaling of a target of the antidepressants that most people are prescribed and this cholinergic nicotinic partial agonist. So where is this happening? We know that 5-HT1A receptors are at least part of one of the targets of serotonin that is signaling downstream of fluoxetine. And so we know that these are expressed both in the serotonin neurons themselves, they're autoreceptors, but also postsynaptically in the hippocampus. So what we've done is to use the same strategy. We made small hairpin RNAs against the 5-HT1 receptor. We infused them either into the dorsal raphe or into the hippocampus. Again, same AAV2, so we're only getting cell bodies, even though the projections from the raphe go to the hippocampus. When we knock down 5-HT1A in the dorsal raphe, we don't do anything to 5-HT1A except on terminals in the hippocampus. And what you can see is that in the animals that have the scrambled shRNA, both a 5-HT1A agonist, that's 8-hydroxy-DPAT, decreases immobility. So does that partial agonist of nicotinic receptors. And once you knock down the serotonin receptor, neither agent is now effective. And really, this speaks to where we are from the 1970s, where we thought of the brain as a big bag of neurotransmitters, where serotonin really was the depression transmitter, or norepinephrine was involved in mania. Obviously, that's not the case. What we're thinking about in general neuromodulators that are released widely throughout the brain that are coordinating the activity of circuits that are important for adaptive behaviors. And so as you look at each of these neuromodulators that are released in response to normal sensory stimuli, in the case of acetylcholine, we know stressful, alerting experiences, that when they actually signal together, they coordinate the activity of circuits, in this case probably originating in the hippocampus, that are important for adaptive behaviors. And as these signals converge, then you get plasticity, and over a longer time period, then you might get aberrant plasticity that will result in these changes, longer-term changes in behavior. So we know that the hippocampus is one critical site because we've actually used AAV2s to change the expression of a couple of different receptor subtypes and molecules in the hippocampus. But is this also the place where this cytosine, this nicotinic agent, or actually nicotinic receptor signaling is occurring? I'm just going to show you a subset of data. We've had a lot more, but this is just to show you that it's not the whole story. So I'm going to show you an incomplete response. So here is one subtype of the nicotinic receptor, alpha-7. It's interesting because it's implicated in some psychiatric illnesses, for example, schizophrenia. It's a weird receptor because it's a homomer. 5 alpha-7s together make one receptor. It's interesting because it's very permeable to calcium, so it's thought to be important for synaptic plasticity. In, for example, the amygdala, which I'll talk about in a minute, alpha-7 is essential in the cortico-amygdala fibers for glutamate-dependent LTP. So it's really been the target of a lot of electrophysiological studies around cholinergic signaling and plasticity. And what you can see is that if we use an agonist, a partial agonist, but an agonist nonetheless, of alpha-7, that we can actually get more time immobile, more time immobile. So this is, again, in sort of the direction of increasing acetylcholine. It's not significant. But an antagonist of this receptor decreases immobility. So this looks a little bit more like the antidepressant-like effect we get of nicotinic blockade. All right, so now, if we look at a poor man's, a biochemist's marker of activity in the hippocampus, generally this is going to be a marker of principal neuron activity because that's where the signal is strongest. What we see is that physostigmine, so again, blocking acetylcholine breakdown, increases activity in principal neurons really pretty much throughout the hippocampus, and we can block that by blocking alpha-7. Again, sort of consistent. But then we get out of consistency because when we start knocking down alpha-7 locally, we don't really have very much of an effect in the hippocampus. We have an effect of alpha-7 to decrease immobility slightly, but it really isn't in any way back to baseline. Here's the increase by physostigmine, and at baseline we have no effect at all. And what I showed you earlier is that if you block nicotinic receptors more broadly, we have a profound effect already at baseline. So this is not the whole story. So what else might be happening? So one of the things that we know is from those microdialysis studies and other studies in terms of inducibility, stress does increase activity of acetylcholine or levels of acetylcholine in the hippocampus. But in particular, the cholinergic inputs to the amygdala are very, very high at baseline, and they stay high with stress. And I don't know if any of you have ever looked at a mouse brain atlas, but did you know that the counter stain to look at the different brain areas is actually acetylcholinesterase? It is. And if you look at the amygdala, the part of the amygdala that really stands out, this is interesting conversations I've had earlier about other nuclei, the hotspot is the basolateral amygdala. And in vivo electrophysiology studies of acetylcholine neuron inputs to the basolateral amygdala, they're extremely high. They're tonically firing at very high rates at baseline. They go up a little bit with a whole lot. So here's an area where activity is high and it stays high. So what happens if we block nicotinic receptors or we have these pharmacological agents that have behavioral consequences to CFOS in other brain areas? So this is, again, an older paper, and what you can see is, and just here's the summary data, if we use either saline or these two nicotinic agents that do different things, the partial agonist and the complete antagonist, that we get a similar behavioral response to these two agents, and the place that we really only see a big similar change in CFOS activity the basolateral amygdala. So the antagonist is pretty good. There's a decrease in CFOS in a number of brain areas. This partial agonist, which again is behaviorally relevant, only really decreases activity here. Why is that interesting? Again, what does a partial agonist do? It stimulates on its own, but if the endogenous neurotransmitter is very, very active, it's going to act to decrease the activity of that normal neurotransmitter. So that's happening in the BLA. So what happens if we go into the amygdala and start to do similar manipulations? Well, here we can put the pharmacological agents just into the amygdala. I'm not going to say basolateral amygdala because we're good, but we're not that good. Basolateral amygdala is still pretty small in a mouse. But if we put it into the amygdala, and here are the placements, we can actually decrease immobility in, here's the tail suspension test, we've done that battery. We can also knock down either the heteromeric or the, or the homomeric nicotinic receptors in the basolateral amygdala. We can show that we can decrease CFOS expression at baseline by either of those knockdowns. So that means, again, tonic signaling through nicotinic receptors is occurring in BLA. And now if we look at behavior, there's something interesting that comes out. One is that across all of these models, here's the decrease in immobility from, in somewhat, in not so much in anxiety-like behavior, but decrease in immobility in these two tasks, increase in exploration, the chronic social defeat. Alpha 7 doesn't look the same. I think that this is doing something different, and I'll come back to this in a second. So there's some effect on this exploration, this anxiety-related measure, much less on these models of immobility and nothing in social defeat. So this homomeric one seems to be really important for this battery of behavioral tests. I think this one reflects data that's come out from Lorna Roll's lab, other labs. This is the receptor that's more important for plasticity. We're looking here at acute effects during a task, so short-term effects. This is the expression of behaviors that we see again and again with acute manipulations using behaviorally relevant antidepressants, but in tests that in fact don't reflect the human consequence of antidepressant use, which only after 3 to 6 weeks of administration. So I think this is what's downstream. I think acetylcholine signaling is downstream of the chronic administration of some of these antidepressant agents that are effective in humans. And that means that the plasticity that might be occurring as a result of alpha 7 signaling may in fact be part of the mechanisms of change in acetylcholine signaling over time. And that's to be done. Okay, so we've got these different effects. We can now challenge with physostigmine. We don't get a complete reversal by these amygdala manipulations, which again goes back to the idea that amygdala is one of the nodes. Hippocampus is involved. There are probably other brain areas, maybe also prefrontal cortex. We've looked there. It's a little more complicated. But we can start to put together under what circumstances is the signaling high, under what circumstances is it inducible. And so this is our current rather ridiculous biochemist's cartoon of the brain. But we're now beginning to interrogate some of the circuit level questions that can be asked about what acetylcholine signaling is doing in each of these brain areas. So for example, in hippocampus, we know that the large majority of, and actually in amygdala as well, the large majority of nicotinic acetylcholine receptors are expressed on GABA neurons. We know that it's probably not selective for one subclass, but we have a lot of different models already from people who have been really bashing the different nice GABAergic subtypes, either in amygdala or hippocampus, showing that, for example, somatostatin positive neurons, VIP positive neurons, and parvalbumin, thank you, PV positive neurons have different roles in amygdala. The same is true in hippocampus. I'm really particularly interested in the idea that some classes of interneurons may be important not for silencing all of their neighbors, but instead for silencing and coordinating the firing of principal neurons so that after the GABAergic activity decreases, the principal neurons are coordinated so that there's feed-forward activity for a long period of time after the GABAergic signaling is relieved. And that's been shown by a really nice a group in Australia who silenced in particular chandelier cells in the amygdala and showed that what you got was a very, very long increase in activity once those chandelier cells were actually relieved of inhibition transiently. One of the ideas is that nicotinic receptors on subclasses of GABAergic interneurons in each of these structures could in fact disinhibit. One possibility is that you have simply a chain of inhibition, so that activating nicotinic receptors on one subpopulation of neurons will disinhibit principal neurons by inhibiting another GABA population. Sorry for the Baroque explanation. But we can interrogate that, and we are now using cell type selective knockdown coupled with DREADs to either increase or decrease acetylcholine in the hippocampus, where we think that the effect of decreasing acetylcholine will be minimal, because we think that stress will actually increase acetylcholine. And here in the amygdala, where we have seen the effect of increasing acetylcholine is minimal, whereas decreasing tone is in fact very behaviorally relevant in mice. So the question is there a way to use the cholinergic input to these two structures to find out whether this is something that is a trait? Is high signaling, we can do this using, for example, the SPECT or PET tracer, is high signaling through acetylcholine in the amygdala a risk factor for developing depression, or a risk factor for susceptibility to stress, or a consequence of stress? And does the dynamic range of acetylcholine signaling here in the hippocampus actually give you more or less resiliency to stress and the development of either anxiety disorders or depressive disorders? And that's something that I would love to be able to work with clinicians to find out. So I am going to stop there, because this is my summary, and I have a whole other thing on aggression, and I'd hate to actually overstay my welcome. But just to go back to the idea that if you start with a human, I think the most remarkable finding of some of these studies is that there is a consistent increase in acetylcholine signaling throughout the brain when people are actively depressed. And if they're remitted, there's still an increase above completely unaffected individuals. And that suggests that it is an area that is ripe for investigation in human subjects. We know that in general, acetylcholine is thought to focus attention. So beautiful studies from many labs, including Martin Sarter's, have shown that what acetylcholine signaling does in the principal neurons, layer 5, decreases noise and it increases glutamate signaling in response to a sensory input. So the timing is absolutely essential. When a relevant sensory input comes in, acetylcholine signaling goes up, and what that does is increase basically the center surround kind of activity in sensory areas of cortex that you would expect is essential for detecting salient signals. However, in the amygdala, we know there's a couple of things that go on. So in fear conditioning, if you increase acetylcholine signaling during encoding, not much more fear conditioning acutely, but extinction learning is slowed, which is, goes along with acetylcholine-induced long-term potentiation of corticoamygdala glutamate signaling onto principal neurons in the amygdala. That plasticity is in contrast to what we see pharmacologically or with the knockdown. We see an acute behavioral response during the task and not learning to respond over time. I think there are multiple things going on. So we would suggest that perhaps too much acetylcholine does focus your attention. It does increase your ability either acutely or chronically to respond to negative information. And our sort of working hypothesis is that's specific for negative information. And we've started to explore this. We've tried to look at what happens if you increase Kay's work. Other work has suggested that, signaling in the amygdala is not only selective for negative valence information. We cannot change positive valence information so far by increasing acetylcholine signaling in the amygdala or decreasing or change the nicotinic receptor control. That's very early. That's why I don't show you these data. May change. But the working hypothesis is that, in fact, what acetylcholine is for is to bias your attention toward those survival-relevant negative experiences, including stress and including dangerous aspects of your environment. And then finally, as I've shown you in both the contrast between what's happening in the hippocampus to what's happening in the amygdala, there are two sorts of signaling, both of which are important in a similar direction. That is that the tonic signaling for acetylcholine in the amygdala is also already essential in unaffected animals to keep their level of responding in these stress-related paradigms high. And at least in a mouse, if you decrease that acetylcholine signal in the amygdala, you already have resilience, you already have more sort of struggle against these stressful stimuli. And in contrast, you have this phasic signaling in the hippocampus. It's induced by relevant information. It's induced by stressful, at least in the mouse, by either social defeat stress or by a physical stressor. And at that point, you have a dynamic to respond in a relevant way to stressful events in your environment. So I'm going to stop there, and I will jump to the acknowledgements so that you can let me know if you have any questions. And I would like to particularly acknowledge the folks in the lab who've done these experiments. And first of all, Barbara Calderon, who's actually at Harvard running a behavioral core, was the first person in the group who made the observation that acetylcholine signaling in the mouse actually was involved in some mouse models of stress-related behavior. And she was the one who uncovered the Janowski papers, which I had not seen. And it's quite interesting why most of us have not seen them, and that is that Janowski, you've probably seen them. And in fact, psychiatrists know them and find them amusing. But what happened was Janowski came up with an entire hypothesis that acetylcholine was the depression neurotransmitter, norepinephrine was the mania transmitter, and therefore a balance between acetylcholine and norepinephrine would determine whether you were in a depressed state or a manic state. And he made this hypothesis right at the beginning of the Prozac revolution. Right as this came out, really the entire paradigm of how does depression work was based on how do serotonin reuptake inhibitors work. And the field went whoop. And acetylcholine has really not been something that's been widely studied. There have been some small clinical trials of, for example, antimuscarinic drugs, primarily by Wayne Drevitz. They've all been quite small and I would say underpowered. Looks like if they were to hold up that it's rapid acting, which is sort of in keeping with the idea that these things are occurring during the time that someone is actively depressed rather than over time. And there have been, unfortunately, there's been a lot of small studies of a nicotinic antagonist in depressed people that showed some efficacy, but one giant AstraZeneca trial failed. So that's never going to go anywhere, which I'm sad about because I do think that there are options for better and more selective nicotinic agents that might be more effective. It's not going to happen. Big failure. All right, so that was Barbara. Reba Ravenstein was a graduate student in my lab and first started using the molecular genetic models, the knockout models, which I didn't show, to look at which receptor subtypes might be involved. Most of the data that I showed you today in terms of the acetylcholinesterase project and the alpha-7 and amygdala beta-2 knockdown were done by Jan Manur, who's a longtime research scientist in my group. And these are the other current lab members. In particular, I want to thank collaborators like Zubin Bagwagar, who's the psychiatrist who did that first study using SPECT tracer in depressed patients. That really was eye-opening for me. Irina Esterlis, I already mentioned, she did the cholinesterase challenge with the SPECT imaging. Ron Duman has been a long-term collaborator. Ralph DeLeon brought AAV2 and the shRNAs to the lab, and then our funders. Thank you.
So just an initial comment. Actually, people thought of using scopolamine as an antidepressant,
but nobody liked it.
Thought of using ketamine, which is now being described as an antidepressant. Both had horrific side effects.
Yeah.
But the problem with scopolamine is there's so many autonomic and other side effects that people couldn't see their way to doing a clinical trial. But I think the jury's still out as to whether it's a rapidly acting antidepressant.
Or not. What's interesting for me is that the nicotinic antagonist studies have never shown rapid acting. The mecamelamine studies that have been done and the one trial with Chantix, with varenicline, which is a nicotinic partial agonist that's used for smoking, both of which showed antidepressant effects in very small groups, had to be given chronically. were given in concert with an SSRI, just as I showed you in the mouse. If you put the two together, subthreshold doses. So in patients who were on Prozac or something like it but were not responsive, if you added either varenicline, which is this nicotinic, it's like cytosine, or the pure antagonist, there seemed to be a better response. What I think would be most interesting is to take a very selective alpha-4 beta-2 antagonist, which would not hit the autonomic nervous system and would be more well tolerated. The problem with mecamylamine is it hits every nicotinic receptor. And that's the drug that was tried in the giant trial that failed. It makes people feel terrible. It backs them up quite thoroughly because you need your autonomic nervous system in order for gut motility to occur. And so you couldn't push the dose. You couldn't actually get up to likely a dose that was getting to significant levels in the brain. I think a more selective agent might, but again, I think that's unlikely to be developed anytime soon.
In the Alzheimer's disease patients, do
Yeah.
they have a lot of incident of depression?
So this is actually very interesting and important. So if we intervene pharmacologically, all of the things I showed you were one injection of physostigmine, and it does what I showed you. If we start to do chronic infusion of physostigmine, we get, as any biochemist knows, basically 5 to 10-fold upregulation of cholinesterase levels and an incredible increase in cholinesterase activity. So these enzymes are extremely homeostatic. Pharmacologically, in Alzheimer's patients, they may in fact, first of all, it's a problem for Alzheimer's. They may have a lot more cholinesterase activity because they've induced this homeostatic upregulation. That's not so good for cognition, but it may be okay for depression because in fact, they may be either at normal levels because of stasis, maybe even less acetylcholine signaling than normal. We do not see that homeostasis with a knockdown, which is very interesting because this is not 100% knockdown. Our efficacy of knockdown by structure is about somewhere between 50 to 60%. Now, whether that means 100% knockdown in 60% of the cells or 60% knockdown in all the cells that express that, we don't really know. And at some point, we can figure out by single-cell-seq. But for now, what we know is that doesn't change over time. And I don't know why. There's no homeostasis in the hippocampus as a result of the AV, which is crazy. So anyway, all of that to say that Alzheimer's patients who are getting tacrine or other cholinesterase antagonists, they get a little tiny bit of boost from it, but probably not very much, and likely because these are biochemically extremely homeostatic enzymes. Yeah?
So what would happen if you deleted the basal layer?
Yeah. Well, so there's a few patients in which that's happened, and they seem to have much less fear, certainly. So fear has always been the primary output that's been investigated for basolateral, or for amygdala in general. If you look at anxiety, there seems to be a very good correlation with activity in the amygdala and anxiety. And so if you look at, for example, Alex Honnold, have you heard of him?
No.
Alex Honnold is a solo climber. He is the guy who goes with no rope to the biggest rocks in Yosemite and climbs them in 2.5 hours because he doesn't take time to put in safety equipment. If you image his amygdala when he is climbing or when he's, or visualizing climbing, because it's a little hard to put an MRI machine on. Yeah, it's actually time when you're in an MRI machine. He has exceedingly low amygdala activity. And that may be partly genetic. It may be actually training. He may have taught his amygdala to do that. But if you look at patients with depression, you see, at least in some studies, increased amygdala activity in fMRI, increased connectivity with the amygdala in resting state connectivity studies, and that may be that may shift with whether they're actively depressed or not, and it's certainly sex differences in that. So the connectivity of the amygdala at baseline is different in men and women.
And it's modulated by hormones.
Absolutely. Oops. Yeah. Yeah.
These mice with the knockouts model for support receptor, ACH1 based with the hippocampus.
Yeah.
Do they have any drowsiness or immobility in general?
Great question. No, nothing that we can see. So if we look at just exploration in an open field, they're fine. If we look at their weight, that's kind of fine. So I don't think there's a big change in arousal. I don't know why. But it doesn't seem to explain the differences in activity in these stressful situations. If they're not in the beaker of water, they're pretty normal looking. Yeah.
So British Titans Group published a paper on Neuro last year. They found that alpha-7 receptor in the hippocampus astrocytes mediates release of E-series,
Yes.
which mediates MBA receptor function.
Absolutely. Yeah.
So here you show that alpha-7, knockdown of alpha-7 also have antidepressant effect. Do you think it might be due to MBA receptor?
So that could be another mechanism, but in our experiments, again, we used AAV2, and as far as we know, it doesn't seem to infect astrocytes. We've looked, I would not 100% rule out a glial phenotype, but as far as we can tell, this is a neuronal phenotype. Alpha-7 is very interesting because it's been shown in astrocytes to mediate or at least be essential for calcium waves in astrocytes. So astrocyte activity seems, in hippocampus and specifically, seems to really require intact alpha-7 function. It'd be pretty cool if there was yet another role for alpha-7 in hippocampus and these behaviors that's mediated through astrocytes. We just didn't test it. And it's quite possible that that's one of the reasons we don't have complete behavioral effects. Maybe we're just targeting neurons, and maybe we should have been targeting glia. We'll do that.
That's great.
Okay. Thank you very much for your attention.